In this figure, we can see the evolution of B cell depleting monoclonal antibodies used in neurology and highlights how antibody engineering has progressively reduced immunogenicity while improving efficacy and safety. Monoclonal antibodies consist of two major components: the FAB region, which binds the target antigen, and FC region, which mediates immune effector function. As monoclonal antibodies have evolved from first generation murine antibodies to fully human and modified antibodies, the proportion of human protein within the molecule has increased, resulting in lower risk of anti-drug antibody formation and infusion related reaction. The first generation monoclonal antibodies include murine antibodies, which were entirely derived from mouse proteins and carried the suffix OMAB. These agents exhibited high immunogenicity, shorter half-lives, and poor clinical efficacy, limiting their therapeutic utility. Subsequently, chimeric antibodies were developed, containing approximately 65% human, 35% murine components, and were designated by the suffix XIMAB. Example, Rituximab, a chimeric anti-CD20 monoclonal antibody. It belongs to this category and became the prototype B-cell depleting therapy. Although not originally licensed for neurological diseases, Rituximab has been widely used off-label in conditions such as multiple sclerosis, neuromyelitis optica spectrum disorder, autoimmune encephalitis, and other antibody-mediated neurological disorders. The second generation includes humanized antibodies, designated by the suffix ZUMAB, in which more than 90% of the molecule is of human origin. Ocrelizumab is a humanized anti-CD20 monoclonal antibody and was first B-cell depleting therapy approved for both relapsing multiple sclerosis and primary progressive multiple sclerosis. Fully human monoclonal antibodies carrying the suffix UMAB represent a further advancement with minimal immunogenicity. Ofatumumab is a fully human anti-CD20 antibody developed for multiple sclerosis, and it offers the advantage of reduced anti-drug antibody formation compared to earlier agents. Third generation consists of monoclonal antibodies with modified FC regions designed to enhance B-cell depletion through improved antibody-dependent cellular cytotoxicity and complement-mediated killing. Ublituximab is an example of glyco-engineered anti-CD20 monoclonal antibody with enhanced effector function. Similarly, Inebilizumab is a humanized anti-CD19 monoclonal antibody that targets a broader spectrum of B-cell lineage, including plasmablast, which are not effectively depleted by anti-CD20 therapies. A key therapeutic distinction exists between anti-CD20 and anti-CD19 therapies. Anti-CD20 monoclonal antibodies such as Rituximab, Ocrelizumab, Ofatumumab, Ublituximab deplete immature, mature, activated memory B cells but spares plasma cells because plasma cells lack CD20 expression. In contrast, anti-CD19 therapies such as Inebilizumab target a wide range of B cell developmental stages, including some plasmablast, thereby producing more comprehensive suppression of antibody producing cells. This difference is particularly relevant in antibody mediated neurological diseases such as NMOSD, where pathogenic antibodies play a central role. In summary, the progression of murine to chimeric, humanized, fully human, and FC engineered monoclonal antibodies have resulted in progressively lower immunogenicity and improved therapeutic effectiveness. Rituximab, Ocrelizumab, Ofatumumab, Ublituximab target CD20-positive B cells, whereas Inebelizumab target CD19-positive B cells, and therefore depletes a broader range of B cell population. These agents have transformed the management of multiple sclerosis, NMOSD, and several autoimmune neurological disorders.